Arsenic and other heavy metals resistant bacteria in rice ecosystem. Potential role in promoting plant growth and tolerance to heavy metal stress. Arsenic and other heavy metals are toxic to plants and humans, and can contaminate soil and water sources. Some bacteria can resist and transform these metals, and also promote plant growth by producing hormones, solubilizing phosphate, and fixing nitrogen. The authors of this article isolated and characterized such bacteria from the rhizosphere of rice and barnyard grass, two plants that grow in arsenic-contaminated paddy fields in Bangladesh. They found that most of the arsenic-resistant bacteria belong to the genus Sphingomonas, and that they could oxidize or reduce arsenic, as well as tolerate other heavy metals such as zinc, cadmium, chromium, copper, cobalt, mercury, and lead. They also showed that these bacteria enhance the germination and root growth of rice seedlings in vitro. The authors suggested that these bacteria could be used for bioremediation of arsenic and other heavy metal contaminated soil, and for improving the yield and quality of rice crops. Bioremediation is a process that uses living organisms to degrade or remove pollutants from the environment. Bacteria are one of the most widely used organisms for bioremediation because of their diversity, adaptability, and metabolic capabilities. Bacteria can interact with arsenic and other heavy metals in different ways, such as uptake, transformation, detoxification, sequestration, or volatilization. These processes can affect the mobility, bioavailability, toxicity, and fate of the metals in the environment. One of the main mechanisms of bacterial arsenic resistance is the Ars operon, which encodes genes for arsenate reductase, ARSC, arsenite efflux pump, ARSB, ATPase, ARSA, regulatory protein, ARSR, and metalloid resistance protein, ARSH. The Ars operon can be found on plasmids or chromosomes of many bacteria. The ARSC gene catalyzes the reduction of arsenate to arsenite, which is then extruded out of the cell by ARSB. The ARSA gene provides energy for the efflux pump by hydrolyzing ATP. The ARSR gene regulates the expression of the operon in response to arsenic concentration. The ARSH gene confers resistance to antimony, another toxic metalloid. Another mechanism of bacterial arsenic resistance is the IO operon, which encodes genes for arsenite oxidase, AIOA and AIOB, and regulatory protein, IA. The IO operon can be found on plasmids or chromosomes of some bacteria. The AIOA and AIOB genes catalyze the oxidation of arsenite to arsenate, which is then released into the environment or taken up by phosphate transporters. The IA gene regulates the expression of the operon in response to oxygen and arsenite concentration. Some bacteria can also methylate arsenic into different organic forms, such as monomethylosonic acid, MMA, dimethylosinic acid, DMA, trimethylarsine oxide, TMAO, or trimethylarsine, TMA. These forms are less toxic and more volatile than inorganic arsenic. Methylation is catalyzed by enzymes such as arsenite methyltransferase, ARSM, or cyclic arsenite methyltransferase, Cas3-MT. Methylation can also occur through a non-enzymatic pathway involving S-adenosylmethionine, SAM, as a methyl donor. Bacteria can use different strategies to remediate arsenic from contaminated sites. Some of these strategies are adsorption. Bacteria can bind arsenic to their cell surface or extracellular polymeric substances, EPS, through electrostatic or covalent interactions. This reduces the bioavailability and mobility of arsenic in the environment. Biomineralization. Bacteria can precipitate arsenic as insoluble minerals such as scorodite, iron-3 arsenate 2H2O, orpiment, as 2S3, realga, as 4S4, or arsenopyrite, FeAs. This immobilizes arsenic and prevents its leaching into groundwater. Bioaccumulation. Bacteria can accumulate arsenic inside their cells or intracellular granules without causing toxicity. This concentrates arsenic and facilitates its removal by harvesting the biomass. Bioleaching. Bacteria can solubilize arsenic from minerals or ores by producing organic or inorganic acids, such as citric acid, oxalic acid, sulfuric acid, or nitric acid. This enhances the extraction and recovery of arsenic and other valuable metals. Biovolatilization. Bacteria can convert arsenic into volatile forms such as TMA or arsine, Ash3, by methylation or reduction. This transfers arsenic from soil or water to the atmosphere, where it can be dispersed or oxidized. Bacteria can also associate with plants to enhance their ability to uptake, translocate, and detoxify arsenic. This process is called phytoextraction or phytoremediation. Some plants, such as ferns, sunflowers, rice, and mustard, can tolerate and accumulate high levels of arsenic in their tissues. Bacteria can help these plants by providing nutrients, such as nitrogen and phosphorus, that are essential for plant growth and metabolism, producing plant hormones, such as indole acetic acid, IAA, gibberellins, GA, cytokinins, CK, and ethylene, ET, that regulate plant development and stress responses, solubilizing phosphate, iron, and other minerals that are important for plant nutrition and arsenic uptake, inducing systemic resistance in plants against biotic and abiotic stresses, such as pathogens, drought, salinity, and heavy metals, modulating the expression of plant genes involved in arsenic transport, metabolism, and detoxification. Some examples of successful applications of bacterial-assisted phytoextraction of arsenic are inoculation of Pteris vittata, Chinese brake fern, a hyperaccumulator of arsenic, with Sphingomonas sp. PV22 increased the arsenic accumulation in the fronds by 53% compared to uninoculated plants. Inoculation of Helianthus annuus, sunflower, a high biomass accumulator of arsenic, with Bacillus sp. AR2 increased the arsenic accumulation in the roots by 35% and in the shoots by 45% compared to uninoculated plants. Inoculation of Oryza sativa, rice, a staple food crop that can accumulate arsenic in the grains, with Sphingomonas sp. SA2 reduced the arsenic concentration in the grains by 53% and increased the grain yield by 40% compared to uninoculated plants. However, there are some challenges and limitations associated with bacterial-assisted phytoextraction of arsenic, such as the availability and stability of bacterial inoculants in different environmental conditions, the compatibility and specificity of bacterial strains with different plant species and cultivars, the potential risks of gene transfer or horizontal gene transfer between bacteria and plants or other microorganisms, the disposal or utilization of the harvested biomass containing high levels of arsenic. To overcome these challenges and limitations, some novel techniques have been proposed, such as phytosuction separation, PSS. This technique involves growing plants in a hydroponic system with a porous membrane that separates the root zone from the nutrient solution. The membrane allows the passage of water and nutrients but prevents the transfer of metals from the roots to the shoots. The metals are accumulated in the roots and can be removed by cutting or washing them. This technique reduces the amount of biomass to be disposed and avoids metal contamination in the edible parts of the plants. Genetic engineering. This technique involves introducing foreign genes into plants or bacteria that can enhance their ability to resist, transform, or detoxify arsenic. For example, overexpression of Rs gene from E. coli in Arabidopsis thaliana increased its tolerance and accumulation of arsenate. Similarly, overexpression of ARSM gene from Rhodopseudomonas palustris in Corynebacterium glutamicum increased its methylation and volatilization of arsenite. Arsenic and other heavy metals-resistant bacteria have a potential role in promoting plant growth and tolerance to heavy metal stress in rice ecosystem. They can also be used for bioremediation of arsenic and other heavy metal-contaminated soil and water by various mechanisms such as adsorption, biomineralization, bioaccumulation, bioleaching, biovolatilization, or phytoextraction. However, there are some challenges and limitations that need to be addressed before implementing these techniques on a large scale. Novel techniques such as PSS and genetic engineering may offer some solutions to these problems.